want to introduce Peggy Ozias-Akins from University of Georgia. We're pretty excited to see what you have to say today, Peggy, about your very difficult problems that you're working on. We appreciate it. Thanks.
Well, thank you, Jeremy, and good afternoon. I'd like to thank the organizers for giving me this opportunity to present our progress on the JGI CSP project we have on Apomixis. You can see the title of the project in red, but I retitled it for the purpose of this presentation. And I think by the end of the presentation, you will see that this really describes our hypothesis. So none of this would be possible without an excellent team of really talented researchers from JGI and Hudson Alpha. You can see many of them here, although I'm sure this is not all of them, but these are the ones that I have communicated directly with, along with Joanne Connor, who is a research scientist here at the University of Georgia, and a colleague of mine who's also worked on Apomixis for many years. So what is Apomixis? Well, it's simply defined as asexual reproduction through seeds, which then enables cloning. So therefore, the offspring of an apomictic plant are essentially clones of the mother plant. And so it doesn't take a huge leap to really be able to recognize the utility of Apomixis. It would have the possibility of preserving hybrid vigor in crop plants. And so if installed in crop plants, basically if there is hybrid vigor demonstrated by the hybridization of two relatively inferior inbreds, then that hybrid F1, if it undergoes sexual reproduction, will undergo recombination and segregation so that the progeny then will not be similar to the F1 hybrid. There will be segregation of phenotypes. However, if that F1 hybrid could be made to be apomictic, then the offspring of that F1 hybrid would be identical to the mother plant and identical to each other. So there are multiple pathways for achieving apomixis, and I'm not even going to present all of these. I'm going to focus on gametophytic apomixis in this talk. But first I wanted to just describe meiosis again in female reproduction in plants. So the female reproduction is occurring in the ovule. There is a single cell that is designated the megaspore mother cell that undergoes meiosis. And this generates 4 haploid products, 3 of which degenerate and 1 of which then develops into the functional megaspore. That functional megaspore undergoes 3 mitoses to form the female gametophyte, also known as the embryo sac, and that is a 7-celled 8-nucleate structure. Within the embryo sac, there is the central cell, which is fertilized by one of the sperm delivered by the pollen tube, and that will then develop into the endosperm. And then there is also the egg cell, which is fertilized by the second sperm, and that will develop into, of course, the diploid zygote and further into the embryo. So in gametophytic apomixis, there are a couple of different forms. One is diplospore, in which the megaspore mother cell, rather than undergoing meiosis, directly undergoes mitosis. And in this case, one of those products will function as the megaspore and will develop into the embryo sac. But now each of those cells in the embryo sac is diploid rather than haploid. As occurs during sexual reproduction, however, the central cell very often in most apomix is fertilized by a sperm cell delivered from the pollen tube, and that's required for endosperm formation. However, the egg cell develops through parthenogenesis into an embryo, and so there is no fertilization of the egg cell. In the materials that we study, the form of apomixis is apospore. In this case, the megaspore mother cell may or may not enter into meiosis, but typically those products will degenerate and there will be somatic nucellar cells adjacent that will develop into the embryo sac, and it may be 1 or more embryo sacs. Again, each of the cells in the embryo sac is now diploid because it developed from a somatic cell. And again, the central cell may need fertilization in order to develop into the endosperm, whereas the embryo is going to develop into, or excuse me, the egg cell is going to develop into the embryo in the absence of fertilization, so through parthenogenesis. So essentially, there are these two components of apomixis, apomeiosis, or apospry in our case, and parthenogenesis, where the egg cell develops into the embryo in the absence of fertilization. So there are a number of challenges for genetic analysis of apomixis. One, which has made this project challenging also, is that apomix, with very few exceptions, are polyploid. Even the diploids are rather with bizarre genomes, and they appear to probably have originated through hybridization as well. The apomix are also heterozygous. Apomixis occurs in species which may or may not have a sexual ecotype within that species. That's the case for one of the species with which we work, and therefore an interspecific cross is necessary to do genetic analysis. In other cases, there may be sexual ecotypes that occur within the species, but they probably are at the diploid level rather than polyploid, although that may not always be the case. But if they are at the diploid level, then perhaps it's going to require either interploidy crosses or doubling of the chromosome number in order to be able to cross an apomict with a sexual. The genetic analysis relies on meiosis, but specifically what we are perturbing in the apomict is the ability to recombine and to segregate in the female. So therefore, we can only use the male for this genetic analysis. However, many apomixes are not obligate. That is, apomixis is not fully penetrant. And so that facultative apomixis, while it could be perceived as an advantage, and it probably is evolutionarily, it does complicate the genetic analysis because it's not predictable. The frequency is not predictable. So in spite of all of those challenges, we pursued genetic analysis in a cross of this species in the genus Pennisetum, which is in the grass family, with another species that had a sexual counterpart with which it would cross. This genetic analysis was done by looking at the linkage of molecular markers with specifically apospry. And one of our early observations was that there was a large number of molecular markers that were completely linked with apospry. And so this led us to the conclusion that there was little to no recombination in this region of the genome. And so we labeled this the apospry-specific genomic region, or ASGR. At the time, we had no clue about the physical size of this region, and so we did embark on generating a physical map using bacterial artificial chromosome fluorescence in situ hybridization. And these back clones were recovered from back libraries with the particular markers that were linked with apospry. When we used them to hybridize to the genome of Pennisetum squamulatum, or dichromosome squashes, we came away with several conclusions. One is that the ASGR actually was found on only a single chromosome, and Pennisetum squamulatum is actually an octoploid. So therefore, it is hemizygous. We did know from our genetic analysis that there was low recombination, but with our physical mapping, we were able to estimate that the size of this region was around 50 megabases. So it was huge. We also determined, which we suspected from the genetic map, but with the physical map, we could actually identify that this region was telomeric. So Pennisetum squamulatum, this just kind of puts it into context with the entire group of Pennisetum. I'm using Pennisetum and Synchris interchangeably. Pennisetum is the older name, Synchris is the newer name. But squamulatum, the apomict that we have studied extensively, occurs within the subclade that also includes Pennisetum glaucum, also now known as Synchris americanus. And this is pearl millet. So it's an important grain crop, particularly in Asia and Africa. This subclade also has a basic chromosome number of 7, whereas most of the other species within the genus have a basic chromosome number of 9. You can also see from this slide the distribution of polyploids. So many of the species are polyploid and many of the species are apomictic. And so for the JGI project, we selected a variety of species across this group, such as we would have a number of the different subclades represented. You can see that we did not select Pennisetum squamulatum, and that's because it has, of all of these, the largest genome size. We had done some genetic analysis, not only with Pennisetum squamulatum, but also with Synchrocyliaris. We had the advantage with the Synchrocyliaris that there was a sexual ecotype actually at the same ploidy level. And so we could do an intraspecific cross to produce a population. Also, we did the backfish in both of these species. And we determined that the ASGR had translocated to a different chromosome when these two species were compared. And also it was in a different chromosomal location, being telomeric in the squamulatum, but interstitial in the Synchrocyliaris. It also was inverted when the two were compared. And we saw this pattern when we used backfish to look at other Pennisetum species, such that the ASGR was telomeric, or in many cases more centromeric, and also sometimes the order of the back clones was different than what we observed in Pennisetum squamulatum. And so obviously there's been chromosome restructuring, not only with the position of the ASGR on a chromosome, but it's even occurring across different chromosomes. And we have looked at this not only with just the ASGR, but also an abundant repeat that at least in the synchrospecies, or the Synchrocyliaris and Synchrocytigerus, is very abundant on the ASGR carrier chromosome. So early on, we were hoping to be able to characterize the ASGR more fully for its gene content by looking for synteny with rice. And this was proposed shortly after the rice genome was sequenced. And so that's quite a long time ago now. We very quickly actually rejected that hypothesis. However, what that research did allow us to do was discover a candidate gene for parthenogenesis. And so this gene was found from sequence that we had generated from an ASGR-linked back clone. This was a baby boom-like gene at the ASGR. Baby boom-like genes have two AP2 domains. They are known to be DNA-binding transcription factors and typically involved in development. We did look at transcription of the ASGR baby boom-like gene in unfertilized ovules of apomix as well as sexual individuals, and found that it was transcribed in the apomix at this stage of development, but not in the sexuals. And then the final evidence for function came from transforming the ASGR baby boom-like gene into sexual species, first with pearl millet. In the case of pearl millet, the transformation was not that easy, but we managed to do it, and then investigated the alteration and reproduction by, again, looking at unfertilized ovules for development of the egg into an embryo. And indeed, we found that the egg would develop into the embryo, and the nuclei in the central cell persisted, which indicated that there had been no fertilization. So we could see this reproducibly. The central cell nuclei persisted, whereas the egg was already developing into an embryo. This gene was also tested in rice and maize, and we saw similar phenotypes. And furthermore, we were able to actually recover haploid plants from pearl millet, rice, and maize. So in all three of these, meiosis was still occurring. So therefore, the egg was reduced. The parthenogenesis of that reduced egg allowed us to recover haploid plants. So we're interested in searching for the apomeiosis component of apomixis from these natural apomix. We have observed that the ASGR baby boom-like genes are highly conserved across species, at least the ones that we have examined thus far. And now the sequence information that we're getting from this project is allowing us to really more deeply look into this. But the hypothesis was that since the ASGR baby boom-like gene was highly conserved and had a function in apomixis, then the apomyosis gene is also likely to be highly conserved, even though both of these genes are in a region where synteny is disrupted. So putting the species that we're most interested in within the context of the entire family, grass family, we are sequencing a number of Penicetum synchronous species, shown here highlighted, but also a couple of species that are from a sister subtribe that are also apomictic. In addition, there are some closely related sexual species that are also already sequenced, such as foxtail millet, switchgrass, and sorghum, which give us then good information for comparative genomics. As far as the evolutionary divergence among these species within the synchrony and the synchro species, the divergence is probably only around 8 million years ago. And within the milleninidae, the two species that we are particularly interested in, the Eurocloa and the Megathyrsus, diverged from one another approximately 12 million years ago. But if we look at the divergence between the two subtribes, that is on the order of 21 million years ago. And so there's been considerable evolutionary time for the divergence of these species. So the species that we have chosen for sequencing include, many of them or most of them include apomix, and we do have two sexual representatives here, one from Megathyrsus and then the other from Synchrosilieris, and this particular sexual line was the one that was used in generating the population, so in crosses with the apomict. This is a difficult project because the genome sizes are large, because these are polyploids. And even though I've indicated the 1N DNA content here, it's really not that simple because these have to be haplotype resolved. And we've now got reference genomes from the ones shown with asterisks here, both haplotype resolved as well as annotated. And so thanks to John Lovell and Joanne, we've been able to look at synteny from 3 of these reference genomes compared with some of the sexuals. One of the sexuals is also the Synchrosilieris. And it's not too surprising that we do see good synteny here among the different chromosomes. But what is perhaps surprising is, and maybe not from our perspective, because we had already known that the Penicetum squamulatum ASGR carrier chromosome was synthenic with sorghum 2, which is also Soteria 2, whereas the Synchrosilieris ASGR carrier chromosome was synthenic with sorghum 7, which is Soteria 6. Looking at the sequences that have now been generated, we can examine this further, and the Synchrosotaceous ASGR carrier chromosome is different than either of those two. It's synthenic with sorghum 8 and 9. The Eurocloa brasantha ASGR carrier chromosome is synthenic with sorghum 3. And interestingly, the Megathyrsus maximus ASGR carrier chromosome is similar, is synthenic with sorghum 8 and 9, similar to the Synchrosotaceous, which is a more distant relative than the Eurocloa brasantha, which is actually in the same subtribe as the Megathyrsus. So even if we use just the ASGR and some of its flanking sequence to try to trace this synteny across the apomix and the sexuals, we really are only picking up the similarity with the flanking ASGR sequence, and this is not really showing the ASGR itself. The ASGR carrier chromosomes that we are identifying are the same that we identified with the full synteny plot. So defining the ASGR, we're looking then more deeply into the homologous or homeologous chromosomes within a particular species. And this is an example from the Penicetum cetaceum. This is actually a triploid apomict, and so there are 3 homologs. The ASGR carrier chromosome is CO3, And when it's compared with either AO3 or BO3, can see that there is an inversion at the telomeric end, but there's also a region where there's no synteny demonstrated on both sides of that inversion. And that can perhaps be better seen here. The synchronous cetaceous has this inversion, which does demonstrate synteny with its two homologous chromosomes, and therefore we're not considering this as being part of the ASGR. And so one of our definitions for the ASGR is that it does contain markers that we've already identified in Penicetum squamulatum or other species, but it's also a region that lacks synteny with its homologous chromosomes or homeologous chromosomes. And so shown in green here is that region as I have defined it, which we've also now been able to narrow it down in two of the other reference genomes, the Urocloa brisantha and the Megathyrsus maximus, although we're still working on the Megathyrsus. And so if we look at the annotated genes within the ASGR of the synchronous cetaceous, it's around 249 genes, which can be narrowed down to about 133 of the ASGR of the Urocloa brisantha. But if we use information across all three of these species and look for genes that are highly conserved and shared by all three of these, we only come up with about 20 genes that are likely candidates for the apomyosis. And then furthermore, the last way that we're using to narrow this down is with a recombinant that was obtained from the population of Synchros ciliaris. So this was the cross between the apomict and the sexual. And recombination does not occur very frequently, but we were fortunate to find this one that retained apomyosis, but it was not parthenogenetic. And so when we looked at the retention of molecular markers and the ASGR baby boom-like genes on this chromosome, we find that those are no longer present. However, there are still markers that indicate a portion of the ASGR is present. And so we're using that portion of the ASGR now to narrow down the candidates even further. And at this point, we have 10 candidate genes that are shared among all three species with a similarity of greater than 90% and still present in the apomyotic A8 recombinant. So finally, in summary, we've found that the Panacea tribe is a rich source of apomix, and particularly the genus Penicetum and Synchros for comparative genomics. We know that apomixis requires at least two genes, one for apomyosis and one for parthenogenesis. Although we can't say for sure there's only a single gene for apomyosis at this point, it seems that there is a single gene for parthenogenesis. But these do have to be maintained in linkage at the ASGR for apomixis to be functional. The parthenogenesis gene, ASGR baby boom-like, that we cloned was shown to function in multiple grass species. And since it does have high similarity across apomix, both within and across subtribes, this does suggest that a gene for apomyosis will be similarly conserved. And so that's the basis of our hypothesis. The JGI now, the CSP project, has generated sequence from 9 of the panacoid apomix that allows us to look for these conserved regions and conserved annotated genes as potential candidates for the control of apomyosis. And if a single gene for apomyosis is discovered and shown to control the trait, then it would simplify apomixis in crop plants if only 2 genes are required. I didn't go into other methods that are being used to install apomyosis, but it's basically 3-gene knockout at this point. And that doesn't work in every species. So this might simplify the installation of apomixis in crop plants. So I will stop there. I'm acknowledging a number of former and current postdocs, students and technicians who have worked on this project, as well as funding sources for support for the project. Thank you very much. If there are any questions, I'd be happy to entertain them.
Thank you, Peggy. Are there questions in the room here for Peggy? Oh, I gotta go up.
Hey Peggy,
Hi.
do you know if there are some like Ts enriched in the boundary of ASGR region?
So there are transposable elements throughout the ASGR and in the boundary. I don't, we haven't really specifically looked for a transposable element family that would be conserved only at the boundary and might have been responsible for its translocation or contributed to its translocation.
Yeah, that's what I thought. That's why I asked this question. Thank you anyway.
Other questions for Peggy before I go down the steps?
Thanks, Peggy. So you found that you think you have one gene that might control apomeiosis across your Ocula and Sencris. That's like what, 20 million years of evolution. Do you think that it's the same allele that's doing it? Do you think that it's like an allele that's kept in perpetuity in these systems and a predisposition to become apomictic, or do you think it's recurrent and independent evolution of an apomeiotic allele at this locus?
Yeah, we don't yet have just one gene for apomeiosis. That's what we're hoping. So that's what we're searching for. But if we look at the parthenogenesis, at this point, we think it's one gene, but we haven't really investigated it thoroughly, and we need to do that also with the new sequence data that we're getting. What has been shown in another apomict, and this is actually in the Asteraceae, is that there were independent evolutionary events that changed the expression pattern such that the gene is not baby boom, it's actually a different gene, but such that a parthenogenesis gene was now expressed in the egg, whereas it was expressed in the egg because of the insertion of a mite. However, the mite insertion location was different in two different apomicts within the same family. And so it looked like it was an independent evolutionary event. We haven't really investigated it to that level in our materials.
And Peggy, that's one of the challenges for these apomics, is that the independent evolution events or in different parts of the phylogeny.
Yeah, and then the other challenge is that we don't just have one baby boom-like gene, honestly. And for example, in Pentacetum squamulatum, it's actually duplicated at the it's duplicated within the ASGR. So we don't know which is the functional copy or if there is only one functional copy. So it gets even more complicated.